Hello, in this video we're going to talk about what it means to be organic and specifically talking about the organic molecules. This video is not going to get in depth with any of the molecules within the four organic molecule classes, but this video is just going to serve as kind of an overview of where we're going over the course of the next several videos. Okay, and so in order to kind of fully understand what it means to be a molecule and what it means to be an organic molecule, I think we have to spend a little bit of time talking about how atoms bond to form molecules in the first place. And so two bonds that are going to be important as we move forward is a covalent bond and a ionic bond. Now we've already in the last video talked about what hydrogen bonds are, and so I'm not going to cover them again. Remember, hydrogen bonds are the bonds that are formed between a hydrogen and a different atom on a different molecule, most notably between water molecules. But in order to kind of fully understand what a covalent bond is, which is really the most important bond when we talk about organic molecules, covalent bonds are bonds that are formed between the sharing of electrons. Okay, and so this diagram or this little animation is going to show how the electrons are actually going to orient themselves in order to be shared. And it's really important to note that it is a strong bond. Covalent bonds are the bonds that are going to form the most multitude of the organic molecules, as we're going to talk about over the next several videos. Okay, and so notice that we're not going to get into too much of the chemistry here, but we have an oxygen atom, and oxygen has 6 valence electrons. Okay, it's got 2 electrons in its first shell, which are not shown in this particular diagram. It's got just the 6 electrons, valence electrons shown. So we've got 1, 2, 3, 4, 5, and 6, and it wants 8, and so it has 6, it wants 8, and so the best way or the easiest way in order for this particular atom to be full in terms of its outer valence electron shelf or shell is to share the 2 electrons that hydrogen has. Okay, hydrogen has 1 electron each, and so this is an electron, this one's an electron, and so those electrons are going to orient themselves to where they form a kind of a shared bond between oxygen and those hydrogens. And so if we watch, the bonds are going to form here. This is a covalent bond. This one's a covalent bond. Okay, these are the valence electrons, and so this is a water molecule, obviously, H2O. We know from our last video that H2O is a polar molecule, and so because these electrons are orienting themselves on this end, this gives this end a partial negative charge, and this negative charge is actually going to kind of repel the partial positive ends of the hydrogen side. Okay, and so you have a partial negative, you have a partial positive. This gives this molecule a polar nature, okay, or a polar property, and these electrons are going to be shared between the oxygen and hydrogen. Again, it's a strong bond, but they are shared. Okay, there is no electron transfer. They are shared between them. Now, they're shared unequally, and in the last video we talked about what unequal sharing leads to. It leads to polarity. Again, these electrons are pulled closer to the oxygen than they are the hydrogen, again giving this a partial negative charge and the hydrogen a partial positive charge. Okay, that is a covalent bond. If we compare that to an ionic bond, this is a relatively weak bond. I shouldn't say it's the weakest. It's a weaker bond than covalent bonds, and it's weaker because it is not an association between two atoms that are sharing electrons, and so they're not bonded and linked together. They are just an association between opposite charges, and so you have a positive charge bonding with a negative charge. Okay, and so what happens? Sodium, which is Na, is going to form an ionic bond with chlorine, and this is just one example. Okay, sodium and chlorine are going to bond. Again, this chlorine atom wants 8 valence electrons, or 8 electrons in its outer shell. It currently has 7. 1, 2, 3, 4, 5, 6, and 7. Sodium either wants 8 or it wants to get rid of this 1. And so it's just going to kick this 1 electron away. It's going to in turn become positive because it removed an electron. Chlorine is going to become negative because it gained an electron. The reason behind the overall charges is, remember that there is a number of protons in the center of these atoms, and so typically the proton number is going to match the electron number, meaning it's going to remain neutral. But the minute you transfer an electron to a particular atom, then all of a sudden it has one greater or one more electron than it has proton, and so it gives it an overall charge of negative, and vice versa. If you remove an electron, it's going to have a one fewer number of electrons than it does protons, and so it's going to become positive overall. And so when we look at the interaction between what happens between the two atoms, you'll notice that this electron was actually transferred from the sodium to the chlorine. Again, it gives the negative charge to the chlorine and it gives the positive charge to the sodium, and the interaction between this positive and this negative brings these two atoms together to form the ionic bond. Okay, so two different types of bonds. It's going to be really important for you to distinguish between those and understand how atoms are bonded together to form molecules as we move into the more complex types of organic molecules like covalent or like carbohydrates, proteins, lipids, nucleic acids, and then into enzymes. Okay. So, what does it mean to be organic? In order for something to be organic, it has to contain carbon, hydrogen, and oxygen. Why is carbon such an important atom? It has to do with the structure of that particular atom. Notice that there is 6, and I say 6, or I highlight 6 because 6 is the atomic number for that particular element. 6 protons, okay, in red, 6 protons. 6 protons in the nucleus, and so there are 6 positively charged particles in the nucleus of this molecule. There are also 6 neutrons in the nucleus of this particular atom, and so that gives it the mass of 12. 6 protons plus 6 neutrons equals 12, and then because it's a neutrally charged atom, there are 6 electrons on the outside of that particular atom. Oop, come back here. And those 6 electrons are going to be the key in understanding why carbon is such a fundamental atom with all, or for all, molecules known to support life. Okay. 2 electrons are going to fill up that inner shell. 4 electrons are going to remain in the outer shell. We know from that last example that molecules, or atoms I should say, typically want 8 total electrons in that outer shell, and so it has 4 spots to bond other atoms to. Okay, and the fact that it can bond 4 other times gives atom or carbon atom a really unique property. Okay, it can form up to 4 covalent bonds with a maximum of 4 other atoms. And so when you have an atom that is that diverse, or can form a diverse amount or a diverse variety of molecules, it really sets itself up to be a substantial part of the molecules, again, known to cause life, okay, or known to support life. Carbon is the 15th most abundant element on Earth, and it forms the backbone of all organic molecules. Okay, carbon is the backbone, or forms the backbone of all organic molecules on Earth and in all life forms. And carbon is really good about forming strong and stable covalent bonds. And again, it can form up to 4. 1, 2, 3, and 4 total bonding sites. Now, carbon can form double bonds, it can form single bonds, it can form triple bonds. It really is a unique atom in that it is very diverse in the number of bonds and the types of atoms it can bond itself to. Okay? Structure results in a tremendously large amount of different molecular combinations, and that's going to be key in the physical number or the sheer number of molecules that are required to support all life in the variety we experience on Earth. Okay, so what are the carbon-based molecules? We know that organic molecules contain carbon, and all organic molecules are going to contain carbon, but they're going to contain carbon in a different ratio. Carbohydrates, like this particular ring, is a molecule that is made up of carbon, hydrogen, and oxygen. Okay, carbon, hydrogen, and oxygen is going to produce a carbohydrate, and carbohydrates are really good at storing short-term energy, as well as structuring themselves to produce long chains of repeating rings like this. And we're going to get into that. Like I said, this is just an intro video to kind of highlight what carbon is and what it does for molecular biology. Second one is the nucleic acids. Okay, DNA is an example of a nucleic acid. It is a helical structure used to store genetic information. The third class of organic molecules are the proteins. Proteins are three-dimensional globular structures that are repeated carbon, hydrogen, oxygen, and nitrogen, C, H, O, and N, sometimes containing sulfur. Nucleic acids contain carbon, hydrogen, oxygen, and phosphorus. And the fourth class are the lipids. This guy is a phospholipid. They are utilized within cells to construct cell membranes, but there are other lipids like triglycerides, and like carbohydrates, they are composed of carbon, hydrogen, and oxygen, but are in different ratios than the carbohydrates. Okay, and there are going to be individual videos on every single one of these classes of carbohydrate, DNA, or nucleic acids, proteins, and lipids in this playlist. However, for the purposes of this video, we're just going to kind of have a general overview of the particular molecules. Okay, so a general overview of the carbohydrates are that they contain carbon, hydrogen, and oxygen, like I just said, and it's very unique to carbohydrates in that they are in a 1:2:1 ratio. An example would be this particular monosaccharide, glucose. You can see that carbon, hydrogen, and oxygen, C6H12O6, is exactly that ratio. Carbon, hydrogen, oxygen in a 1:2:1 ratio. It doesn't matter if it's a single glucose molecule or a single glucose ring. It doesn't matter if it's a double ring, like a double glucose ring, okay, which would result in maltose, or it doesn't matter if they're really, really long chains of these repeating rings. It is still going to be a carbon, hydrogen, oxygen in a 1:2:1 ratio. Okay. The monomer, and this is a new topic or a new idea that I'm throwing at you in this video. Monomer is the single subunit of a particular molecule. Okay, and so the monomer of carbohydrates is monosaccharide, meaning single sugar. Dimer, di means two, so double sugar, and polymer is multiple sugars. So you have a single sugar, a double sugar, and a multiple sugar. And when I'm saying single, double, or multiple, I'm talking about the number of rings. So if this ring right here by itself was a standalone molecule, it would be a monosaccharide. The minute we add 2, it becomes a disaccharide. And then when we have 3 or more, it becomes a polysaccharide. Now, some of the specific examples that I would encourage you to know is the monomers would be glucose, fructose, and galactose. The disaccharides would be sucrose, lactose, and maltose. And polymer, the specific polysaccharides, would be glycogen and starch for the functional polysaccharides and cellulose for the structural polysaccharides. Now, I know I threw a lot of sugar names. Notice that all of the sugar names, or most of them, end in OSE. Okay, that's going to be a suffix that you should key in on. If it is a sugar, it typically ends in ose. Now, starch and glycogen are obviously different than that, or don't really follow that rule, but for the most part, if it is a sugar, it's going to end in ose. Glucose, fructose, and galactose are the monosaccharides. Glucose obviously being the simplest, most important, most studied, most understood monosaccharide because glucose is in fact the energy source for the cells in your body. Disaccharides, sucrose, that is table sugar, that is the sugar that most people probably think about when they think of sugar, or at least the form of sugar they probably think about when they think of sugar. Lactose is the milk sugar. It is a sugar that is produced by mammals and is placed in milk to help add energy to that particular substance. And maltose is a sugar that is found typically in plant roots and or plant seeds, and it's typically used to energize developing embryos or developing seedlings. The polysaccharides can be either functional or structural. Glycogen and starch. Glycogen is the functional polysaccharide found in animals, and starch is the functional polysaccharide found in plants. Okay, it makes sense. When you think about a potato, what is the first thing that typically comes to mind when you talk about the nutrient availability within that vegetable? It's typically thought of as being very starchy, and in fact potatoes are the portion of that particular potato plant where the starch is stored. Okay, and so plants produce starch, animals produce glycogen. Plants also have a structural polysaccharide, and that is in the form of cellulose. Cellulose is the structural polysaccharide that orients itself, like this diagram is showing you, into kind of crisscrossing lattice structure, and that's going to give rigidity to plant cell walls. Okay, now animals don't have cell walls, and so we don't need cellulose. We have cell membranes around our particular cells, and that helps to keep our tissues more fluid. It allows for more range of motion. Plants don't need to have motion, and they need to have rigidity, and they need to withstand wind and stuff like that, and so they require cellulose. Animal cells do not. Lipids, there are two lipids that we're typically going to talk about in detail. There are a couple other of other forms of lipids that we're going to talk about just kind of as ancillary information, but the two main types of lipids would be the triglycerides and the phospholipids. The triglycerides are going to be a molecule that contains a glycerol head in green and is going to contain 3 fatty acid tails. So you can see the glycerol head that is bonded to the carboxyl group end of a fatty acid, and the fatty acids are just repeated CH2s, unless we get into saturation versus unsaturation, which will be a later topic. But glycerol, 3 fatty acid tails, and the triglycerides are really good at storing energy for the long term. Okay, so triglycerides are things like fats and oils and waxes and steroids and cholesterol and molecules like that. Phospholipids are the other type of lipid. Okay, and so phospholipid also contains a glycerol. You'll see that this is the glycerol, just like this was the glycerol, but it only has 2 fatty acid tails, and the third fatty acid tail, to be completely detailed, was actually bent up this way and actually attached itself to a phosphate, and we get this phosphate head. And the fact that we have a phosphate attached kind of to this third tail, which has been kind of folded up into the head, into part of this hydrophilic structure, it allows for the molecule to be amphipathic. And so what that means is you have a hydrophilic head and hydrophobic tails. Now, triglycerides do not have the phosphate, and so they don't have the same physical properties as the phospholipid. They are fully nonpolar, and they are fully insoluble in water, and they are fully hydrophobic. The phospholipids have a very unique structure and a very unique function in that they have both polar and nonpolar regions, and that's really good and sets them up for producing membranes in that bilayer structure that we'll talk about later. Steroids, you can see there is more of a ring structure. It might resemble a carbohydrate, but it is not carbon, hydrogen, oxygen in a 1:2 to 1 ratio. In fact, it is a 4-hydrocarbon ring that is not in a 1:2 to 1 ratio. And so if you see something that looks like this, you might be tempted to think that it's carbohydrate, but in fact, it is a lipid. Okay? If we focus on the actual tails of the phospholipid and triglyceride, you will notice that sometimes the tails are completely straight, like this one, and sometimes the tails are bent. This is a zoomed in version of what this diagram over here is showing you. Notice that these are more zoomed out, and so you can't see the individual carbons and hydrogens, but you can see the bends or the kinks in these particular fatty acids. When a hydrophilic or a hydrophobic fatty acid tail is completely saturated with hydrogen, notice that there is a hydrogen at every location and bound to every carbon in that fatty acid chain. It is said to be saturated. And so what are we referring to when we say it is saturated? We're saying that it is saturated with hydrogen. Okay? If the carbons are completely saturated with hydrogen, they cannot form a double bond between two carbons. Okay, and so the difference between a saturated and an unsaturated fatty acid is that when we strip some of the carbons off, you form the need for the carbons to form a double bond with each other, and anytime there's a double bond, it's going to form a kink or a bend in that fatty acid. The more bends, the more unsaturated it is, and the more unsaturated it is, the more flexible it gets. Okay, flexibility is really good when we start talking about fatty acids and the importance of unsaturated fats in your diet and the detriment that saturated fats have to your particular diet. Okay, the more unsaturated and the more double bonds a particular fatty acid has, the more flexible it's going to be. And so here are a couple examples, these being all completely saturated, these being unsaturated. Okay, unsaturated, remember, or see, there is double bonds that you can see in these particular fatty acids. The more double bonds there are, the more fluid and the more bends or kinks in that particular chain. Okay. One thing to key in on, though, when you look at the particular fatty acid, especially if you're having to identify a structure like this from a group of others, is the carboxyl group. And so you can see in this kind of pinky shaded region the COOH. The carboxyl group, we are keying in on COOH. COOH, COO, is the carboxyl group. Okay, and carboxyl groups can be seen in this particular region as a COO bond and then an OH, and can also be seen here. I know you can't really see them as well, it's not as close, but that is still a COOH. COOH is the carboxyl group, and all fatty acids are going to have them. All of them have them here, all of them have them here, these two have them right here. Proteins, the third of the four classes, contains carbon, hydrogen, oxygen, and nitrogen. Nitrogen being the difference between carbohydrates and lipids. Okay, sulfur can also be seen and is commonly incorporated as well. And again, just like in carbohydrates, we have monomer and polymer. Monomer is the single subunit of a particular molecule, and so the single subunit of the particular protein are the amino acids, otherwise known as peptides. Okay, amino acid can be seen here. This is a diagram of an amino acid, and amino acids make up proteins. This is a protein in the quaternary or a tertiary structure, and we're going to talk about that in a little bit. But proteins or polypeptides are another name for proteins, are just repeated peptides or repeated amino acids into a chain and then folded into secondary, tertiary, and quaternary shapes. Okay, long singular or multiple chains are the polypeptides. And proteins, unlike carbohydrates, which typically only have one function, I mean they have two, they're either structural or they're functional, proteins have a whole bunch of particular functions. In fact, you have hormones, you have enzymes, gas transport. Proteins are antibodies, or are a main component of your antibodies and help with the immune response, immune system. Proteins can also be structural, and proteins can help transport things around cells, and they can also transport things across membranes. Okay, proteins are a very diverse class of organism or of molecules, but these proteins all have one thing in common, and they have the same amino acid monomer, and they are all focused into or all linked together and bent into similar looking structures. But another example of structure dictates function, and so they all have a slightly different structure. Some amino acid differences. Notice that the sections in red are different and the sections in black are the same. This was the amino acid that we looked at the last slide, and so this is one section, this is one section, and this is one section, and they are all attached to the center carbon. Okay, and so what are those sections? You have an amino group, sometimes just called the amine group. You have a carboxyl group. Okay, again, that is that COOH that we saw with the fatty acids. C double bond O and an OH makes the carboxyl group. And then you have the R group, which stands for radical. The radical group is the only thing within this entire amino acid that is different between all amino acids on the planet. Okay, so this is kind of the X variable. It is the difference. And so you can see the carboxyl group is the exact same on every amino acid known to man. And the amino group is the same on every amino acid known to man. And both amino acid and carboxyl group are attached to the center carbon in every case. It still has a hydrogen or a standalone hydrogen attached to that center carbon at every case. But it has a different and a very unique radical group on every amino acid, which makes every amino acid different. Structure dictates function. If you have a different structure, you're going to function differently. And so these are essentially the letters that all amino acids, if we were using them as an analogy, like letters, the proteins that were produced by sequencing those particular letters together would be the words. And so it's very easy to understand or very easy to comprehend that different words can be made with the same alphabet. Okay? These amino acids are the alphabet. This could be analogous to like A, B, C, D, E, F, and so on. Okay? And so by linking A, E, F, you produce a different protein than B, A, D, and so on. Okay, and so we can link these amino acids together in a specific order to produce a specific protein to have a specific function. Maybe this one is a hormone or becomes a hormone, and maybe this one becomes some kind of structural protein. Nucleic acids are the last of the four groups. Nucleic acids are contained or help contain or are designed to contain hereditary information or heredity, right? We store our DNA in the form of deoxyribonucleic acid. In fact, that's what DNA stands for. And so nucleic acids are really good at storing genetic material. Okay, and so how is DNA structured? DNA is just like any other organic molecule. It contains carbon, hydrogen, oxygen. It also contains nitrogen and phosphorus. And it too has monomers and polymers. Monomer, remember, is the single subunit. The single subunit of nucleic acids would be the nucleotides. This is an example of a nucleotide. All nucleotides have three groups, 1, 2, and 3. And when you link those individual nucleotides together, it produces a nucleic acid. And there are two types of nucleic acids that we talk about in this class. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. Now, you'll notice that these two names end in "-ose", and so those, if you remember, are sugars. Deoxyribose is a particular pentose or a particular 5-carbon ring structure that the phosphate and the nitrogenous base bonds to. Okay, so this would be in blue the sugar. It is a pentose sugar. Okay, 1, 2, 3, 4, and 5 carbons make up that pentose sugar. Pent meaning 5. This number 3 would be the nitrogenous base. It is a nitrogen containing base, and this over here would be the phosphate. Okay, the phosphate of a nucleotide is always attached to the 5th prime carbon. The nitrogenous base is always attached to the 1st prime carbon, and the next nucleotide is always attached to the 3rd prime carbon. Okay, and so if we understand this nucleotide structure, then we can go over here to the actual nucleic acid, which is multiple nucleotides. Notice that this is a nucleotide and this is a nucleotide, and those two individual nucleotides have been linked with a covalent bond, and it's actually a phosphodiester bond, but don't worry about that now. We're going to talk about that in a later chapter or a later topic, but it has been linked, and so we have taken two of these individual nucleotides and we have linked them together with a bond here, creating a covalent bond. Okay, they are sharing electrons just like we talked about in the first slide, and all we do is we link nucleotide to nucleotide to nucleotide to nucleotide, and then we just repeat that over and over and over and over. And notice that there are several nitrogenous bases. You'll see that there are T's and A's and C's and G's, and so those nitrogenous bases can be A, T, C, G, and if it's RNA, they can be U for uracil. This is obviously a DNA molecule, and so we only have A's and T's. T bonds with A, C bonds with G. We'll talk about that in a little bit, but that is a kind of a general overview of the nucleic acids. The last thing we need to talk about before the end of the video is how all of this plays into this idea of metabolism, and you'll notice that there is a pretty crazy looking diagram in the back of this slide, and you'll notice that there are a lot of molecules that we will eventually talk about, like we just talked about fatty acids. There are all kinds of interactions. Notice that there are also kind of arrows that are pointing from one thing to the other, and they're all kind of interrelated somehow, and so that's what metabolism is. Metabolism is controlled by enzymes, which are protein, polypeptides, and because all of these enzymes and interactions all kind of interrelate and interconnect, metabolism by definition is the sum total of all enzyme-catalyzed reactions in the cell or organism, and so all of those enzymes working in concert with each other, producing products that each other needs and that each process needs in order to function correctly, is what metabolism is. Okay? Enzymes are employed at every step in order to break down and build up molecules more efficiently. Enzymes are quaternary proteins. They are three-dimensional folded chains of amino acids, and there's a video that we'll watch in a few videos in this playlist that will kind of talk you through what enzymes are in a deeper level. Okay, for now, just know that they are molecules that are used to build up and break down other molecules. And so how are those molecules actually built up and torn apart? That gets us into anabolism and catabolism. Anabolism is the building up of larger molecules, and so this is obviously the production or a process of producing proteins. In this particular example, we know that it is building proteins because we have individual amino acids. Remember, amino acids have their amino group, they have their carboxyl group, and they have their R group, radical group. The radical group is the only group that is different. And in anabolism, it is a condensation or a dehydration synthesis reaction. In fact, it's called condensation or dehydration synthesis because we are releasing water. We're releasing a hydrogen and an oxygen. I should say we're releasing a hydrogen and an hydroxide ion, an OH. And when you put an OH and an H together, you get H2O, which is a water molecule. Okay, and so how do we do that? We identify the OH from one carboxyl group, and we identify a hydrogen from the amino group, and when you cut those two structures off of those two amino acids and link them together, you produce a water, and because you cut off a water, they can form a peptide, which is a type of covalent bond because they're sharing the electrons. You're forming this covalent bond between the carbon and the nitrogen. This carbon was the carbon of the carboxyl group. This nitrogen is the nitrogen of the amino group, and it forms this new bond which creates a 2 amino acid long chain called a dipeptide. Again, di meaning 2. If we were to add a third one, then it would become a polypeptide, which is protein. Okay? Now this process happens in all organic molecule classes. We link individual monosaccharides together with dehydration synthesis or a condensation reaction. We link individual nucleotides together to form a nucleic acid with a condensation or a dehydration synthesis reaction. And the only way, really, to just summarize this, to produce bigger molecules is by removing water. And so if we have an amino acid, and I know this is a very non-detailed drawing, but we have one amino acid and we want to bond it to the next amino acid with a peptide bond, we have to pull out an H2O. And we want to link another amino acid, and so we have to pull out an H2O. And then we want to link another or amino acid, and so we have to pull out an H2O. Okay? And so we pull out H2Os every time we want to link a small unit or a small single monomer to our growing chain of whatever it is we're growing. Catabolism is the exact opposite. Catabolism is the breakdown into smaller components. And so how does that work? It requires enzymes, just like anabolism did. But catabolism, we're using the enzyme to actually split a larger molecule apart. And so you will notice that there is an oxygen that is bound to both carbons. This would be a carbon, this would be a carbon. And in order to break the bond between these oxygens and carbons, we have to add water in. This enzyme is going to grab a water molecule, it's going to split the water molecule up into an OH and an H. Okay? It's going to give the H to one of the O's, it's going to give the OH to the carbon, and now you're going to see the carbon that received the OH has the OH, and the oxygen that was bound to the carbon now has its individual hydrogen, and because they are bound to each other now, they let go of each other and formed two smaller monosaccharides in this case, glucose and galactose. Because they are two individual monosaccharides, we have split this disaccharide, which is a larger molecule, into smaller components, smaller molecules. That is catabolism. Okay, that's where we're going to end it. If you have questions or concerns, bring them to class. Otherwise, have a good night.